So thank everyone for being here. And so what I'm going to do today is use half of my talk to present a problem, and that is the problem that we are trying to crack. And then towards the end, I will show you some data to exemplify how we are addressing one aspect of this problem. And if there is time, towards the end, I will tell you the sort of a bit of the follow-up. Now, imagine that you have to go through this path. By the way, this is my commute in Munich. No complaints. So what you're going to be doing is using optical flow. That means a full visual motion, visual field motion. And therefore, you will be able to locate a path and know that there are obstacles around, and you will also be able to regulate as well perhaps the speed and the accuracy of your movement and so on and so forth. Now, let's start doing some experiments. Let's assume that you cannot see anything. It's pitch dark. Can you do this? You will have a bit of a difficulty, but you can still do it. And the reason is the following. You now are not going to use visual cues, but to some extent, you will start using mechanical cues. And so the path is hard, and it's harder than the surrounding field. And therefore, what you could use is somatosensation, that is how the ground feels on your feet, and therefore can walk along this path that is harder, and you navigate this path pretty well as well. Let's do another experiment. Let's see you're floating. Can you do this? The answer would be, well, no, because I have no way of telling that there is anything there. There are trees, there is a sort of a path that is hard, and so on and so forth. Now, imagine now another experiment, which is even harder for someone to sort of be able to know what's going on. Imagine you're floating in the ocean, and it's pitch dark, and there is a current. You actually don't know you're moving. So the current is moving you, but you can't tell. And you may say, but we have our vestibular system that will be able to tell that at least our head and the body attached to it is moving. But it's not the case, because the vestibular system will adapt pretty quickly. And if you imagine, for instance, you're on a moving train and you close your eyes, very quickly you don't realize that you're moving. So basically, if you are suspended in a medium that is moving you, in principle, you can't tell that you are being moved. The reality is that some animals can, in fact, not only tell that they are being moved, but also knowing in which direction they are being moved. Question is how? And the main problem here is that these animals, which normally would use whole visual field motion for navigating or guiding themselves, will have to transform what it is intrinsically an exocentric reference frame to one that is endocentric. That means they have to feel their own body and then see how they can use the information that the body is getting to navigate. And this essentially is a computational task. Now, I will now show you the actual problem with a fish. This is a little fish you see up there. It's a larval zebrafish. This fish is inside a seamless cylinder filled with water. This is the side view, this is the top view. The fish is about 1 week of age. It's very, very small. Now, what's going to happen now is that there's going to be flow of water, and you will see water is flowing in this direction. The fish is actually now swimming against the flow in a directional manner. You say, well, yeah, sure. The fish is seeing that it's being moved by the flow and tries to keep up with the flow. Now, the reality is that this fish is in complete darkness. It cannot see anything. The question is, how can it tell both that there is flow and determine direction of flow so that it can engage into this counterflow behavior? This, by the way, is called rheotaxis. So the question here is, how can the fish tell that there is flow and determine direction without using exocentric reference frames? What the fish does is using an endocentric reference frame using exclusively mechanosensation. So, again, the problem is essentially a computational problem. And the way we address it, in part, is by using David Marr's sort of levels of understanding of a computational problem. One is the actual computational problem. So what is the goal of the computation? In a sense, is asking the animal, why do you care? Why do you do this? This is one level. Second level is the algorithmic. In other words, how the input and output are represented, in principle, in the brain, so that the fish can engage in a motor reaction. And the third one is the implementational. Like, what is the physical substrate? What is the animal using to extract relevant mechanical information from the environment and then engage into this behavior? So what we are going to do now is discussing a little bit the logic of rheotaxis and then concentrating later on this level, the implementational level of Marr's understanding of a computation. So what we do is the following. We take a fish, again, we put it into a cylinder, a seamless cylinder, and then we apply flow. And what you see here is the fish, top view, flow, the fish starts moving. And what we can map in 3 dimensions is not only when the fish swims in bouts, in which direction it's swimming, and also color code the timing. Now, flow here is laminar. And this is important. And the reason why it's important is that laminar flow has a perfect mathematical solution. That means that once we know how the water is flowing in the cylinder, we can directly relate the statistics of the stimulus, mechanical stimulus, with the actual behavior of the animal. And this is very powerful when you try to address a computational problem, being able to actually relate quantitatively input and output. Now, what happens is that flow in a cylinder that is laminar has this parabolic sort of form. The reason, and we are not going to discuss the physics of this, is because of something called boundary layer. That means that water flowing right next to the edge of the cylinder will flow very, very slowly. Within the boundary layer, there's going to be essentially no flow. And as you progress from the boundary towards the center, flow will become faster and faster until you are at the very, very center where flow is fastest. And then as you go to the other direction of the cylinder, you again describe the exact same parabola, but in the other direction. Now, the fish itself also produces a boundary layer because it's a solid object immersed in water. So around the fish, there is also going to be this kind of degree of velocity profile. What happens is that if the fish is located in the center of the cylinder, the flow on one side and the other side are going to be the exact same velocity. But as the fish moves from the center to the periphery, the flow on one side is going to be faster than on the other side. That will immediately create a local vortex around the fish. And this vortex is 3-dimensional. In other words, there is a vortex around the plane, the azimuth, and a vortex that is orthogonal, right? The fish is actually going to start using this mechanical information to detect flow and determine the direction. Now, the question is how? The main thing here is that the fish needs to break symmetries. This is essentially symmetric. The fish will have to extract inhomogeneities that have a pattern within this flow to be able to perform the task of rheotaxis. So, in other words, to understand this computational problem, we need to understand the steps of symmetry breaking. What are the 3 symmetries that need to be broken? One is called range fractionation. The fish will have to get rid of information that is not useful. So it fractions the range of information and it extracts only what it needs. The second, well, in other words, what the fish is going to be doing in this case is getting rid of one plane of mechanical information and saving, in a way, one of the 2. Therefore, what it is doing now is reducing the computational problem into a two-dimensional problem rather than a three-dimensional problem. It only needs to detect vortex along one plane or across one plane. Second level of symmetry breaking is that the system needs to be bidirectionally sensitive so that it can tell in which direction flow is going, but it has to break that symmetry as well, so each direction is informed to the brain or transmitted to the brain separately, so that the brain eventually can integrate information on one side of the animal and the other side of the animal. And by working out how this vortex flows around the body, the fish will be able to break the problem or solve the problem of determining flow direction, well, detecting flow and determining flow direction. So what we are going to do now is looking at the implementational part, exclusively concentrating on the periphery. How is the fish acquiring information, breaking symmetry at the periphery and acquiring information? So the fish uses a system that is called the lateral line. This is a collection of mechanosensitive organs that decorate the entire body of the animal. These are mechanoreceptive organs again. The sensory cells are mechanoreceptive cells that we also have. Those are located in the ear, so called hair cells. This system is absolutely awesome. Unfortunately, we lost it, evolutionarily speaking, in the transition from water to land. And basically, the animal can use this system to detect mechanical information around the entire body. So if there is an object, for instance, it will be able to detect the presence of the object. If there is a threat, it will be able to swim away from the threat using mechanical cues. It can also hunt using this system by detecting mechanical disturbances in the water by a potential prey without vision. So when my computer science friends tell me about augmented reality, tell them about the lateral line, which I think is much more awesome. Now, these hair cells project from the body of the animal, so they're in direct contact with water, and so water movement will mechanically stimulate these cells. Now, these cells come in two flavors within each organ. Half of them, if you look at the apical domain of these cells, will have a projection, that is the mechanical apparatus, that forms this horseshoe-shaped structure pointing in one direction. Whereas the other half have the same structures, but pointing in the exact opposite direction. So the epithelium of the organ is what is called, within the plane of the epithelium, bipolarized. Now, this allows this organ, by virtue of how these organelles, apical organelles, act, to detect flow in two directions. And that is because if you mechanically push this organelle in this direction, you will stimulate these cells, whereas if you push them in the opposite direction, in reality, something happens, but for the purpose of this talk, we'll say nothing happens. Whereas for these ones, the exact opposite will happen. If you push them in this direction, nothing will happen. If you push them in this direction, they get stimulated. So the organ can, in fact, detect flow along this axis. If you push them in the orthogonal axis, nothing will happen. Therefore, the system at the periphery already is breaking some symmetries and doing this range fractionation, because flow in every other direction will not stimulate the organ. The organ will now be stimulated only by flow in this direction. But there is a problem. There is directional ambiguity. The organ itself is not going to be able to tell in which direction flow is coming. So the system will have to break this symmetry as well. So this is the representation. Now, one of the motivations to do this work was because of the hair cells. So we are born and die with the same hair cells. If we lose them, we become deaf, and that is forever. In the case of a fish, that's not the case. If we wipe out the entire hair cell population, within 2 days, it regenerates everything. So one of the motivations as well behind this project is to try to understand hair cell regeneration, because we are also interested in sort of real world problems and see if we can, discover something that would be important for regenerating hair cells in humans. One thing that we discover is that by using light imaging, is that hair cells in neuromast developing pairs. So what you see here is a pair of hair cells and a hair cell progenitor. Progenitor is going to divide now, producing 2 hair cells. These progenitors appear only at the top and bottom of the organ, so the organ is compartmentalized. Not compartments in the Drosophila sense, just for the cognoscenti, that's not what I mean. Let's say they are territories. And what happens is that the siblings, we discover as well, are going to be facing one another. In other words, one of the cells born from this one division will be facing one direction, its sister the opposite direction. And as progenitors appear and more pairs of hair cells are born, you get this sort of pattern in that you have an axis of mirror symmetry across the organ. How does that happen? How is that every time 2 hair cells are born, they face one another? And that's always the case. They never make mistakes. So this is the question. And so we decided to sell our soul to the devil and do single-cell RNA sequencing to try to understand whether we can understand this production of 2 hair cells that are facing one another by looking at their transcriptional profile. Do they express different genes? So we did this, but among other things, we also, this was a rich, or remains a rich source of information to try to understand how hair cell progenitors are produced and then hair cells mature and become functional. Now, one of the things we knew is that there is a transcription factor called atonal, some of you may know from flies, ATO in vertebrates, that is expressed very strongly in hair cell progenitors. And so in the progenitors of the progenitors, which are epithelial cells, is not produced. And as hair cells mature, the expression goes away. So we could identify which cell we were looking at and the expression profile, just exclusively looking at the profile, temporal profile of atonal. And therefore, we can locate within this representation where the progenitors were present, basically, were located. Then we decided to focus on another gene, among others. We analyzed quite a few. This one is called EMX2, it's a transcription factor as well. And we discovered that it was expressed in the same cells as atonal, but as things progressed, it didn't behave the same way. It started to remain expressed in some cells, some hair cells, but it went away in some other hair cells. Question is, why? And this is an actual combination of nuclear stain of a neuromast, a transgenic line that expresses GFP in hair cells, Dhabi for seeing all the nuclei, and what we see is EMX2 expressed, this is the protein, in only 3 out of the 6 hair cells, which made sense because we predicted that from the single-cell RNA sequencing data. Now, there was another candidate that we looked at, that is the receptor Notch, intercellular communication receptor, and in particular Notch1A. And we had already a suspicion that Notch1A in Notch signaling via Notch1A might be involved in this sort of pattern of EMX2 expression. So we decided to mutate it. And so what we obtain is now a situation where every hair cell in the neuromast expresses EMX2. In other words, atonal is necessary for the production of progenitors, and somehow between here and here, Notch1A is going to repress EMX2 expression in one of the 2 siblings. What is interesting is that now, if we look at the actual polarity of the hair cells in a wild-type neuromast, we see half of the hair cells pointing one direction, the other half in the opposite direction. In a Notch1A mutant, where all the EMX2, all the hair cells express EMX2, now every hair cell points in the same direction. That tells us that EMX2 may determine the direction of polarization of the hair cells. And if that is the case, what happens if we eliminate EMX2 completely? What happens is that now every cell points also in the same direction, but opposite to the direction of the Notch mutant. That means that EMX2 is precisely the it kicks the cascade that is going to determine the direction of polarization of the hair cells. and it's a symmetry breaking via notch between the siblings that is going to determine that one points in one direction, the other one in the opposite direction. And so the model is precisely that one. There is a signal, and that signal generally is the death of hair cells that is going to trigger the production of a progenitor. Victoria hasta la muerte. 2 hair cells are produced. There is a communication via notch between the 2 siblings, and that is going to make one remain EMX2 positive and the other one EMX2 negative. Determine the polarization of the hair cells. And as the cells keep on progressing, this is going to be stabilized, and this is essentially going to allow the organ to be bidirectionally sensitive. And then something else happens, which is that neurons are going to be able to recognize the polarity of the hair cells and stabilize synapses with half of them, each neuron here with half of the hair cells, but these cells, to which this one marked neuron stabilized synapses, are all pointing in the same direction. That means that these synapses, specific synapses with one polarity, is going to break the other level of symmetry, which is the vectorial ambiguity of the organ. And this information is now going to be transmitted to the brain via separate channels and integrated in the brain so that the third level of symmetry will be broken. With this I finish, but not before mentioning the funding for this work. Essentially a combination of NIH funding through the BRAIN Initiative. I'm part of a large consortium based at Harvard that is essentially trying to tackle behavior generally, and our contribution is rheotaxis, but also by the Aerospace Agency in Germany. And then there are other projects in the lab that I will have to wait for another occasion that are also generously funded by other institutions. Thank you very much.